just beginning to get into the actual mechanism of how these receptors work, okay? Now, these are all adrenergic receptors that we're going to be looking at the details of the mechanism. And so they're all slow receptors. So let's kind of just jump back here and kind of clarify. We've got 3 types of receptors that we're working with, mainly in the autonomic nervous system. We've got cholinergic receptors, but there are 2 types. There are nicotinic and muscarinic. And then we've got adrenergic receptors, which are pretty much all slow receptors. The cholinergic receptors, the nicotinic are fast receptors. The muscarinic are slow receptors. The adrenergic receptors are all slow, okay? But there are several subtypes. We talked about them at length last time. And so we're going to talk about some of the mechanisms that differ between those different adrenergic subclasses. So let's start off here by looking at the mechanism for the alpha-1 receptors. And just to refresh ourselves, we said alpha receptors were kind of the most common, usually had an excitatory response at the tissue level. And I'll talk a little bit later on about differentiating between the tissue response, be it excitatory or inhibitory, and the actual effect on the second messenger, okay? Because we can have a second messenger, we can have an increase in second messenger, which brings about an inhibitory response and vice versa. So there are 2 separate things, what this receptor does at the tissue level and what its effect on its second messenger is. Alrighty, so let's start off here with alpha-1 receptors. And so we can kind of you can write in your notes that this is an alpha-1 receptor. I usually have students kind of get confused about the way that I explain this. And it's usually because we explain it in one membrane. So you're going to see the receptor here in one membrane, but it's actually just a schematic representation. We need to be remembering that these receptors are actually on completely different tissues, right? So beta 1 is going to be in the lungs and the kidneys and in the heart. Beta 2 is going to be on the respiratory tract, so lungs, smooth muscle. Beta 3 is going to be on adipose. So while I'm explaining them here on the same membrane, remember that they're actually going to be found on different tissues. So alpha-1 receptor here in this membrane, it's a metabotropic receptor, a slow receptor, so it's coupled to this G protein. This happens to be a G stimulatory protein. That's not always the case, so kind of make a note of that. After this hormone or neurotransmitter binds to this receptor, it's going to have a configurational change. The G protein is going to dissociate that alpha subunit off. It's going to go downstream in the membrane, and it's going to activate an amplifier enzyme. And we kind of talked about this loosely. We just said that it was X amplifier enzyme. And here we're going to add some detail to what that enzyme is. So in this case for alpha-1, the enzyme is called phospholipase C. And it's going to activate this enzyme, and that enzyme is going to cleave another molecule in the membrane, okay? The other molecule is PIP2, okay? Phosphatidylinisetol bisphosphate, otherwise known as PIP2. And so phospholipase C cleaves PIP2 into 2 separate compounds, IP3, which is inositol triphosphate, and DAG, which is diacylglycerol. And you don't have to be concerned too much about the actual names. On an exam, you'll probably likely see the abbreviations. So PIP2 is cleaved into these 2 compounds, IP3 and DAG. They're actually both second messenger. IP3 is more involved in the second messenger role. And these 2 molecules are going to enter the cell, and they're going to do some different things that lead to a cellular response. IP3 usually is involved with calcium release. And DAG is usually involved with activating another enzyme that phosphorylates some type of protein and generates a response that way. Okay, so I'm going to walk through that series of events just because repetition is so important for these processes so that you can really understand them. Let's walk through that, that sequence of events a second time here. So norepinephrine, epinephrine, either at a junction or as a hormone binds to this alpha-1 receptor. It has a configurational change on the G protein that it's coupled to, such that the alpha subunit floats off, dissociates, moves downstream in the membrane, activates this amplifier enzyme, phospholipase C. Phospholipase C is what's going to generate or bring about our second messenger. So it's going to cleave PIP2 into IP3 and DAG, IP3 will generally be more involved in the second messenger pathway. It's going to enter the cell. It's going to go to the endoplasmic reticulum. This is the organelle where calcium is stored, and it's going to encourage calcium release. And in things like muscle, release of calcium from the endoplasmic reticulum is going to generate muscle contraction. So that's how we're going to generate a cellular response, okay? But we're not too worried about what that response is at this level. The other part of the second messenger response is going to be DAG. Diacylglycerol, again, enters the cell. It's going to activate some protein in the cell. Again, the details here are not that important, but just understand that these are our two second messengers, and these are going to lead us to our cellular response. Okay, so that's alpha-1 receptors. That's the mechanism that only applies to alpha-1 receptors, okay? Alpha-2 receptors and all beta receptors use an entirely different amplifier enzyme and an entirely different second messenger. So really, really important distinction to make. So let's talk about the alpha-2 receptors and beta receptors. Now, again, I'm illustrating them here on the same membrane, but we want to remember that this is actually happening on different tissues, on different cells. And so for the sake of clarity and simplicity, you'll see those mechanisms in the same membrane, but remember they're happening at different tissues. So let's walk ourselves through what happens with alpha-2 receptors first. So alpha-2 receptors are coupled to a G inhibitory protein. Okay, really, really important. And so their action is going to be determined by the fact that we're actually inhibiting the second messenger here. Not the second messenger, the amplifier enzyme, but the amplifier enzyme, which eventually results in inhibiting the second messenger. So the alpha subunit, again, dissociates off, but it's going to inhibit this amplifier enzyme. And the amplifier enzyme here is adenylate cyclase, and that is going to inhibit the release of cyclic AMP, okay? And so we're going to kind of pause here and look at what happens to the beta receptors. So the beta receptors are coupled to a G stimulatory protein. So while they're using the same amplifier enzyme and eventually the same second messenger, the action on that amplifier enzyme and second messenger differs between alpha-2 receptors and all three of the beta receptors. So the beta receptors are actually going to stimulate adenylate cyclase, which is going to increase the amount of second messenger produced. Here, the second messenger is cyclic AMP, one of our nucleotide derivatives that we've talked about, and that is going to generate our cellular response. Cyclic AMP is going to enter the cell, it's going to activate protein kinase, which will phosphorylate some protein in the cell and bring about our cellular response. And I want to make it clear here, this is a point that usually students have a hard time with. So alpha-2 receptors are coupled to a G inhibitory protein that is going to cause an inhibition of adenylate cyclase, which is going to decrease our cyclic AMP, but that will also generate a secondary response. That will also generate a cellular response, okay? So don't get in the trap of thinking that because I'm inhibiting my amplifier enzyme, I'm inhibiting second messenger, that I'm going to be ceasing cellular response. No, this is just going to create a different cellular response than if I were activating second messenger and activating the amplifier enzyme. But both scenarios will generate a different response respectively. Okay. All right. So that's kind of the mechanism that alpha-1 receptors use, alpha-2 and beta receptors use. Let's pause here and see what questions you may have. Okay, so I'm going to go over the beta receptors again. So someone's asking me to clarify the beta receptors here. Oops, okay. So beta receptors coupled to a G stimulatory protein, meaning that the alpha subunit here dissociates off and activates adenylate cyclase. Adenylate cyclase is what produces our cyclic AMP, which is our second messenger. This is the compound that actually goes into the cell and starts stirring things up that generates our cellular response. So in this instance, it's going to activate protein kinase, When we see the word kinase, it's going to add a phosphate. So it's going to phosphorylate some type of protein, and that is how we get our cellular response. The alpha-2 alpha-2 receptors, on the other hand, couple to a J inhibitory protein. So when that alpha subunit dissociates off, it's going to inhibit adenylate cyclase, which is going to decrease cyclic AMP. And the important part here is that when we inhibit cyclic AMP, we create another type of cellular response. Remember, we talked about this covalent regulation, that when I add a phosphate, I'm going to activate a protein that may do one thing, but if I'm not adding a phosphate, it's going to do another thing. So both options would generate cellular responses. They're just going to be two different cellular responses, okay? All right, so although they're using the same second messenger, they can bring about two totally different cellular responses because one is inhibiting and one is activating. Okay, hope that added some clarity there. Okay, excellent. So let's look at this table here, really, really important table. I want you to be able to, or I think it's helpful for you to be able to recreate this table on your own, okay, from your own understanding. So you should definitely have a good understanding of this table. So the receptor type, what tissue we're going to find that receptor on, what the relative affinity is for norepinephrine and epinephrine, right? So beta 1, beta 3 prefer have an equal affinity. Beta 2 prefers epinephrine. All the alphas prefer norepinephrine. You're also going to want to know the signal transduction mechanism. So whether it's going to activate its second messenger and what the second messenger is, or it's going to inhibit that second messenger. And then lastly here, you're going to want to be thinking about the effect on the tissue level. So we said all alpha receptors are excitatory, and that means that they're usually going to bring about activative responses on the tissue level. So things like vasoconstriction, bronchoconstriction, just really exciting the tissue, increasing heart rate, increasing respiratory rate. And when we think about beta 2 receptors, they're usually going to be inhibitory at their tissue. So they're going to cause bronchodilation or vasodilation, and it's going to be more of a relaxing inhibitory effect at the tissue level. I also want to point out here, again, something I mentioned earlier, but do not confuse what it does to its second messenger with what it does on the effector organ or the tissue level. So for example, alpha-2 receptors are inhibitory to their second messenger, but that actually generates an excitatory response, okay? Beta-2 receptors are activating their second messenger, but that generates an inhibitory response. And we can see here that although all beta receptors activate their second messenger, which is cyclic AMP, that brings about totally different responses. One, because they're actually on different cells, right? And two, because activating this second messenger and inhibiting the second messenger are bringing about different events in the cell. Okay. Another kind of helpful table here, this is more of the clinical importance of understanding these second messengers and these adrenergic receptors. And so what pharmacologists can do, and what we do in modern medicine, is we kind of manipulate our understanding of these receptors. So for example, when we think about, let's take isoproterenol. This is actually an asthmatic medication, and it's basically mimicking the response of norepinephrine and epinephrine binding to beta 1 and beta 2 receptors. And because that generates a bronchodilation or a relaxation of the smooth muscle in the respiratory tract, that's going to be ideal for someone who has asthma, right? Asthma is this blocking of the airway, inflammation of the airway, constriction of the bronchioles and of the airway. And so in this type of patient, we want to kind of manipulate our understanding of these receptors, and we can agonize or mimic that response, and that will be ideal for that patient. Okay, we can think about this with the cholinergic receptors as well. So pilocarpine is an example of a medication that's usually given to treat dry mouth, dry eyes, glaucoma, and it's actually going to agonize muscarinic receptors. And if you think about the function of the parasympathetic system and the muscarinic receptors, they're going to increase glandular secretion. So increase salivary secretion, increase lacrimal secretion of the eyes. And so that's going to be, again, the response that we want in these patients. So we can kind of take our understanding of these receptors and use that to either agonize or antagonize, so either mimic or block some of these receptors and generate the responses that we would need in some of these patients.